Okay, Team Bio, welcome to your screencast on a leaf cross-section. So yesterday or 2 days ago, we talked about the structure and some of the functions of the chloroplast. And today, we're zooming out and talking about where do we find chloroplasts? Well, often they're in leaves. What's the structure of a leaf? So, hence the screencast. Okay, imagine you have a leaf and you cut the leaf in half right here. And then you discard this part of the leaf. You don't need it. You take the leaf and you rotate it, keeping it on the same plane. Just swing it around this way and then spin it 90 degrees this way. And you will have a leaf that looks something like this. Now you're looking at it on end or via the cross section. So here is the stem. This is the top of the leaf. Here's the bottom. For all of you that thought, oh, leaves are so narrow and thin, they can't possibly have more than one layer of cell, you're wrong. There are multiple layers. And today's screencast is going to be about those layers. So imagine we take a zoom in box of this view of the leaf, and then we zoom out from it to here and here, and then we examine the layers within a leaf. So that's today's screencast. Get ready. When you come back to school after March Intensive, I'm going to give you prepared slides of leaf cross sections. You have to bring them into focus and then label them using your notes as a guide. It'll be pretty fun. Okay, so let's get right to it. The very top of the leaf. This is not a layer of cells, but it is a layer of wax. It is called the waxy cuticle. So the leaf produces a layer of wax. It is a protective layer, and it's doing a huge amount of protection against dehydration. So the waxy cuticle is hydrophobic, and so water is not going to easily pass across it. And so this waxy covering on the outside of the leaf prevents the leaf from losing water. But the waxy cuticle is not a cell layer. It's actually produced by the upper epidermis, which is our first cell layer. So the outside of your skin is called the epidermis, and the outside skin of a leaf is also called the upper or the epidermis. How convenient. So this is a protective layer. It's helping secrete the waxy cuticle, and it has a very striking boxy-like shape. So it will be relatively easy to identify underneath the microscope. Okay, directly underneath the upper epidermis is a layer of cells that's very important. It's called, I don't think I want to do an arrow, the palisade mesophyll. You'll notice that there are tons of green little dots in the palisade mesophyll. And every green little dot is a chlorophyll organelle. Oh, no, that is not a chlorophyll. That's wrong. Sorry, it's early. It's 9:20, or sorry, I can't even know the time. It's 6:20. No, these are not chlorophyll. They're chloroplasts. Okay, so the palisade mesophyll is chock-a-block full of chloroplasts. There are also, these other structures that you can see here that are vacuoles. So, what's the chloroplast doing? Photosynthesis. What is it producing? Glucose. We need somewhere to store the glucose in the cells that it's being produced in before it's exported to the rest of the tree. And so we store that glucose, that extra glucose, in the vacuole. So the palisade mesophyll, very tall layer, also boxy, and it is the primary site of photosynthesis. So think about the location of the palisade mesophyll relative to the leaf and the energy source for photosynthesis. I'm not going to tell you, but I think you can figure it out. Why do you think the palisade mesophyll is where it is? Okay, the next layer of cells is a little bit less organized. It's purposefully got lots of little air pockets within it, and it is aptly named. It is called the spongy. That's a G. Mesophyll. So just like a sponge has lots of little air pockets creating like a space for compression, and it gives it this kind of the squishy, spongy texture, the spongy mesophyll has these little boxy cells and then air pockets in between. And we're talking about the air in just a second. But back to the structure of these spongy mesophyll cells, they also have chloroplasts in them. So I wonder if I can circle one. They have chloroplasts, and they also have vacuoles. I think I'm having a hard time circling it, but you get the point. Here's a vacuole as well. So the spongy mesophyll is the secondary site of photosynthesis. Most of the photosynthesis is happening in the palisade mesophyll, but there's also some happening in the spongy mesophyll. They're equipped, they have the stuff they need, the chloroplasts and the vacuoles to store the glucose that they make. Okay, let's move on down here so we can talk about gases. Okay, guess what this is? Yes, you guessed it right. It's another epidermis, but this one is the lower epidermis. Same functionality as the upper epidermis. It's protective and it helps, so it secretes the wax cuticle. We also have a wax cuticle down here. Same thing, protective and a hydrophobic barrier preventing the cell from being dehydrated. However, if you look over here, there's something interesting going on. The lower epidermis is not completely contiguous. There are some gaps or breaks in it, and these are specialized cells. These two pink cells that I've drawn here are called guard cells. These two, they're both called guard cells, but I'm not going to draw a thing across the whole thing because I'm going to have to draw other things going in and out in a second. Okay, the whole structure is called a stoma, not to be confused with the stroma within the chloroplast. The stoma the plural is stomata. They just throw in a random extra T. I'm not sure why. And so the stoma is equal to 2 guard cells. Oops. And these guard cells, yes, as you might expect, they can swell up, and when they do, they close this opening, and then they can also shrink down and create a little gap into which gases can move into and out of the leaf. So this is how the leaf is breathing and allowing for gas exchange to take place through the stoma. The way that these structures swell up and then shrink back down, yes, you guessed it, it's via osmosis and the pumping of ions across membranes. If you're interested, you should look it up. Okay, so what is coming into and out of the cell? Well, not the cell, the leaf. The leaf is performing photosynthesis, and you know that the raw ingredients for photosynthesis are CO2 and H2O. So CO2 has to come into the leaf. It is in higher concentration outside of the leaf than it is inside of the leaf because we're constantly consuming it in the process of photosynthesis. So it's going to flow down its concentration gradient from an area of high concentration outside to an area of lower concentration inside the leaf. H2O is actually coming from a different structure, which we're going to talk about in a second. But it's at higher concentration. First, I'm not going to talk about that. First, I'm going to talk about oxygen. So oxygen is being produced in photosynthesis. So if we look at the concentration of oxygen inside the leaf relative to outside of the leaf, we see that it's at higher concentration. So oxygen, when the stoma are open, is going to flow from inside of the leaf to out this way. And water is also actually going to be at higher concentration. Water vapor is inside of the leaf, and so when the stoma are open, water is going to be flowing out as well. The reason why the stoma would stay open is because they want to bring in CO2. But the reason why they would close is they want to prevent the leaf from being dehydrated, from losing too much water. Plants have this interesting challenge on really hot, dry days. They want their stomata to be open so they can get CO2 in, but they also need their stomata to be closed so they can avoid being dehydrated and losing too much water. So we're going to be talking about some adaptations plants in desert environments have made to be able to continuously bring in CO2 or keep releasing CO2, and conserve water. But that's for another time. But you should know that trade-off exists. Okay, so those are the gases going in and out. Now, how does the water get here? Well, let's move over here and examine this structure here. This is called the vascular bundle. I think I had it on 3. This entire little wheel-like structure is called the vascular bundle. And it is composed of 3 different cell types. First, it has the bundle sheath cells. These cells surround the vascular tissue, and they kind of keep it organized and in place. They are kind of like the walls of imagine this is this is the vascular system of a plant. It is kind of analogous to the arteries and veins that run through your body. So the bundle sheath cells are keeping these in quotes arteries and veins. You're going to learn their real names in just a second. Organized in a bundle here, and you can see these if you look at a leaf. These are all vascular bundles here. So the veins in a leaf are examples of these vascular bundles. So when we look at a leaf in cross-section, we are going to encounter some places where we've cut through this piping and we can see it in the face of the leaf. Okay, so there are 2 different structures within the vascular bundle. The first is the xylem. The xylem is responsible for bringing water up from the roots of the tree into the leaves of the tree, and it's doing so via capillary action. So water is pulled up from the roots to the leaves in these tiny, 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 tiny little polar tubes where the plant doesn't actually have to exert any energy in order to bring it up to, maybe 200 feet above the ground. Okay, so this is how liquid water gets into the leaf, and it is going to eventually evaporate in the leaf. And so we are going to have some gaseous water vapor along with our oxygen and our CO2, which are the other primary gases within the leaf. Okay, the secondary structure in the second structure in the vascular bundle is called the phloem. And the phloem is responsible for bringing, I guess I'll make it orange. Sugar water. And transporting that sugar around the entire plant. So the leaf is where most of the photosynthesis is happening in a tree, for example. And mostly it's happening in the palisade mesophyll, but it's also happening in the spongy mesophyll. And so tons and tons of glucose is being produced in this structure. And we need to get it around to the rest of the plant because cells that are in old bark sections, for example, that are not green are not performing photosynthesis, and the roots are not performing photosynthesis. So the sugar water is going to be exported to the rest of the cell, and the cell is going to use it for energy and growth. Okay, so I think that's it. In your next screencast, we're going to really dive into the structure of the chloroplast and talk about the thylakoid membrane system, what all of the different proteins and photosystems that are embedded within it, and how it actually does its job of capturing sunlight energy and then using that sunlight energy to drive photosynthesis. So that's all, but now you know where this is all happening. See you in class on Monday, Tuesday after March Intensive. Hope you have a fantastic March Intensive. Can't wait to hear about them when we get back.